Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in part C in CSIR NET exam. So this is one question, one example question. Let me take a color. Let's take it. Okay. So two homologous proteins were isolated from a psychrophilic and thermophilic conditions. Okay, so psychrophile and thermophile. Psychrophile denoted with P, thermophile with T. The purified proteins were subjected to denaturation, protease digestion and circular dichroism. Following observations were made. So firstly, you need to read the questions couple of times to understand it very clearly. We are talking about two homologous proteins. These are homologous proteins, same type of proteins, isolated from two different type of organism in a psychrophilic environment from a thermophilic environment. So you need to have a clear idea about psychrophile and thermophile, otherwise you cannot even think of answering the question. So if you don't have the idea about it, you should leave. But if you know that psychrophile means informed of cold temperature and thermophile means hot temperature. Okay, thermophile means those organisms who lives in the hot climate as an optimum temperature, and psychrophile means cold climate. So now you know the difference between a same homologous protein in two different conditions, in very cold region as well as in very hot region. And then we check that protein with the help of denaturation, protease digestion and circular dichroism. Three separate techniques. Following observations are made after using these techniques. So techniques like denaturation, protease digestion and circular dichroism, you also need to know what these techniques actually do in order to answer the question. Denaturation means we are going to open up the structure of the peptide into the primary sequence, or even not even to primary sequence, at least the tertiary structure should be broken down into the secondary structures. Protease digestion can help us understand how the different fragments of amino acids generated, and circular dichroism, CD. Circular dichroism helps us to understand the change in the protein conformation by any means. So now the observation, the CD spectra of P and T proteins are identical. Okay, the question says that among these statements, when you read these statements, these are true statements that we got. Okay, these are experimental data. The CD spectra for both P and T are identical. And why not? Because homologous proteins, their amino acid composition is 95% identical. Yeah, that's also fine. T and P are equally susceptible to proteolysis in the presence or absence of reducing agents. Fine. T has higher midpoint of thermal denaturation than P. The reason, so this is the last thing that is unique. Okay, so among these four statements, if you read them carefully, the CD spectrum is same between the two, so it's nothing new about it. The amino acid composition is 95% identical, nothing new about it, almost very identical. T and P are equally susceptible to proteolysis, so nothing new. But the only thing that differs between T and P, thermophile and psychrophile, is that the higher midpoint of the thermal denaturation is for the P. So for psychrophile the thermal denaturation point is low. For thermophile, the thermal denaturation point is high. What is thermal denaturation point? It means a particular temperature at which the denaturation, we can say midpoint, means half of the proteins get denatured at a temperature. That is the thermal denaturation point. So this midpoint of thermal denaturation is higher for T, means the thermophiles. To denature thermophile proteins, we need to apply more heat compared to denaturing psychrophile. So if you know this, the protein from psychrophile, right? So if you know this, there is something about the protein structure of this thermophile which makes the thermophilic protein more strong, more heat resistant compared to the psychrophilic protein. Now what may be the cause? The four statements: altered secondary structure, increased number of disulfides in the T, increase in water of hydration, increase in number of salt bridges. So these are the four statements. Among the four, you need to answer the correct one. Okay, so which among the four is correct? Now think about it. So ultimate thing is, so all this question, if you read it very carefully, this whole question is of a huge time wastage to read through, the circular diachromosome, protease digestion, denaturation. We don't even need any of that. The whole question depends on one thing. We can break this question in simple form. Like you have two polypeptide sequence, two protein sequences. One protein sequence, upon adding heat, denatures fast and the other denatures slowly. Now what makes the other protein denature slow? That is the question, essentially the same question like this one. But this question is showcased in a different way to make and sound more difficult. So now you know that the altered secondary structures have nothing to deal with the thermal stability of a protein. The thermal stability of a protein obviously must be linked with the bonds that are present in the protein, right? Now which bonds are responsible to produce and provide a protein more thermal stability? And this is something you should remember, you should know. If you don't know the property of these different bonds and their features, you cannot answer that. Okay, so the question is asked from the different bonds that are present in the protein. And we know that among all these interactions of disulfide interaction, water in the hydration is not important, but among two interactions, disulfide bridges as well as the salt bridges, we know among these two, salt bridge interactions provide more structural stability in high temperatures. So what you can say is that the salt bridge interaction is most important, which is present, like more number of salt bridge interactions are found in T, thermophilic protein, compared to that of the psychrophilic protein. So that should be our answer, option number 4. So if you see this very carefully, this question will consume 5-6 minutes of your time. I took more time because I need to break everything and explain. But even if you read through this question, it seemed complex because it is built complex, it's made tricky, but actually depends on the simple idea of the thermal denaturation of a protein. Okay, once you know that, you can clearly complete this question. Okay, now let's go to the second question. Binding of two ligands to their binding proteins were investigated. Following binding isotherms were obtained. Now this is just a graph. Okay, first of all, the question is not that big, so little information is provided. The idea is about the binding of two ligands to their respective binding proteins. Okay, so one is the ligand A, another one is the ligand B, and what we are looking, the binding of ligand A and B with their target protein, and the comparison we see in the bound concentration of the ligand A and B and the bound by total number of this ligand available, the protein available. Now basically, so we have bound in Y and bound by total by X axis, means the percentage of this A and B that is bound. So think about it, this question is about how the protein is binding to the receptor. Okay, so the ligand is binding with the protein and the binding of ligand with the protein based on the bound and unbound state. So this is what we need to understand. Okay, now if you look at very carefully, for A, you can clearly see the bound by total concentration, the bound by total amount. So as we are going, the percentage of the bound is increasing, so the total amount of bound proteins are decreasing at this point, slowly decreasing with for the protein A, right? So let's say there are 10 bound out of 20 total, so 50% are bound at a stage. Okay, at this point 50% are bound, and the total amount of protein bound are also provided. So if the amount of protein is decreasing, the protein's binding capability is also decreasing. You need to understand the relation between X and Y. In this Y axis we have only the concentration of the proteins, and in the X axis we have the percentage of the bound protein. So the bound percentage, literally if you write bound percentage in the X axis, it will be much easy for you to solve this instead of thinking of bound by total. It's nothing but a bound percentage. So if we are decreasing the total bound protein, the bound percentage is also decreasing. Okay, so decreasing in the total protein, the bound percentage is also decreasing, and it's decreasing gradually here, okay, in a fixed value for A. But for B, at the very beginning, where the percentage, the bound state is huge, while most of the proteins are associated, like ligands are associated with the protein, then even the percentage of the bound is altered, is increased, still it is not going to change the bound state of the protein towards ligand, and it will change when the percentage bound decreases, I mean increases, percentage bound increases with time. Okay, so it is going to tell us one thing. It's going to tell us this interaction between this ligand and the protein is non-covalent interaction. If it is a covalent interaction, then you will never obtain a graph like this, this kind of a graph. We'll always get a straight line graph. We're not going to get the curved line graph. That means the interaction is not, it's not non-covalent interaction. Okay, so the options are given: A is obtained with an oligomeric protein, B is obtained with monomeric protein. There is nothing about the monomer or oligomer of a protein that can change this interaction. It's totally depend on, like you can see, the B is obtained with protein with positive cooperativity. A and B were obtained by the same protein at two different temperatures. The profile of B is not possible. Now we know one thing is that the profile of B is possible because the profile of B also has a relation. It's not like that they don't show any logical relation. There is a logical relation. It is when we are talking about the ligand binding with the protein. Okay, now sometimes what happens, the ligand can bind with protein. Let's say one ligand binds with a protein with a certain affinity, and in the second time the second ligand bind with the protein with little more affinity, and then the third ligand bind with this protein with even more affinity. So if you plot this kind of graph, you will see this kind of bent curve for that, and this kind of binding is observed for the oxygen's binding affinity towards hemoglobin. So the more oxygen is bound, the more affinity the oxygen gets towards the hemoglobin to bind with it, or towards the RBC to bind with it. So that's why this kind of binding is an example of cooperative binding or positive cooperative binding. So in the graph, if I show you the graph, this is just an example of positive cooperative binding, where you can see the percentage bound state. As the percentage bound state is increasing, I mean the bound content, the more bound they are, the percentage bound they are going to increase over time. Okay, while the negative cooperativity suggests it's going to decrease. Okay, if let's say negative cooperativity means, obviously if one ligand binds, that will allow the next ligand to be also released from that, and it will get more difficulty for the next ligand to bind with the protein from the first one. So this process is known as the negative cooperativity. So we can easily find this out with this sketched plot by reading it, by reading the graph. So this question totally depends on the reading of the graph, your skills of reading of the graph. And the best way to reading a graph is always try to look what is present in the x-axis and what is present in the y-axis, and find out the relation between the x-axis and y-axis. Okay. So let's So let's move on to the third question now. It says hydrogen bonds in proteins occur when two electronegative atoms compete for the same hydrogen atom, like one donor hydrogen and one acceptor. Now both donor and acceptor are fighting for the hydrogen, then only the hydrogen bonds form. True. We also know that for H2O. Okay, so any cases where the hydrogen bonds are present, H2S. Now that the angle theta between donor and acceptor of hydrogen bond was determined from the large family of X-ray structures of proteins as shown below. Now normally what we know is for water we have a structure like this, H2O. Now for this structure there is a particular angle created. This is the theta angle that we are talking about, but not for the water molecule here. We are talking about a protein, an amino acid we are talking about here. Okay, so the hydrogen bond that is present between that, and X-ray structures of a protein showcase that. Okay, and the value for the theta is listed in the x-axis in all this graph, and the number of hydrogen bonds present is present in the y-axis. Okay, so this is the data set that is given, and by looking at this data set we need to find out which among these three graphs represents the correct order of finding out the arrangement of theta. Okay, so the angle theta between donor and acceptor of hydrogen bond was determined, and what we are seeing here is that so the angle is written in the x-axis. So from 100 to 200, a number of hydrogen bonds based on how much degree of angle is created. So what they want to know, the relation between the angle, the degree of the angle, and the number of hydrogen bonds. That means whether there is any relation between the angle that is present between the different atoms, between the different bonds, or with the number of hydrogen bonds or not. If there is any relation, that's what they want to know. If you don't know, you cannot answer the question. You need to know this idea that for the hydrogen bond to occur, these atoms most of the time need to be present in very close proximity. So in this case, this is water. We are not talking about any hydrogen bond here, because there are second water molecule can form a hydrogen bond between themselves like this. This will be the hydrogen bond, okay, hydrogen with oxygen, one donor, one recipient. But this one is the normal structure of the water, okay. So the donor acceptor of the hydrogen, so donor and acceptor of the hydrogen, if you think about the relationship here, so the theta that we are looking here is between the donor and acceptor of the hydrogen bond. So this, in this case, the donor is this one, this is the donor, and this is the acceptor, okay, and the hydrogen bond formed with an angle there, okay. So that angle we need to figure out. This is called theta in this occasion, okay. So what we need to figure out, among these three graphs, which represents the value of theta best. Now as per the graph number 1, as the degree of angle increases, the number of hydrogen bond decreases. As per the second graph, there is an optimum value for the angle that has the maximum hydrogen bonds, and both the extremities will have lower number of hydrogen bonds. And the third graph represents, if we increase the angle theta, more hydrogen bonds will form. Now this is a make or break question. That means either you know the answer or you don't know. And in this case I will explain that yes, the thing is, this third one is the truth. So the number of the more angle they have, the higher chance of more hydrogen bond formation. So if you look at here, the strongest will have this maximum angle in theta form there, okay. So the more value of the theta, the higher the value of the theta, the more hydrogen bonds you can see. So for the angle of 150 degree, less than 160, little more, 180, little more, and 200 is the maximum number of hydrogen bonds formed. So this is something of a direct question asked in a different manner, okay. Now let's talk about this question. In a 30 residue peptide, the dihedral angles phi and psi have been determined by one or more methods. When their values are examined in Ramachandran plot, there are 4 different statements provided for the same. Now when you read these questions, this question is very common. Actually they ask this question almost every single time for CSIR NET exam. At least one of this kind of question from Ramachandran plot regarding phi and psi angle. I already told you that it's very important for you to understand and remember the phi and psi values for different sort of secondary structures, for alpha helix, for beta sheet, for right-handed, left-handed helix, and so many things. So to answer this question, you need to know what exactly provided by the Ramachandran plot. You need to know what Ramachandran plot provides us. It says not possible for phi and psi values to be distributed in the helical as well as the beta sheet region, okay. Second thing, possible that phi and psi values are all in the helical region, although circular dichroism spectrum studies indicate beta sheet conformation. So the phi psi values provided from this, inferred from this Ramachandran plot shows that the protein is going to be helical, but the circular dichroism spectra is going to give us the information of the protein as beta sheet. Possible to conclude that the peptide is composed of entirely D amino acid. Not possible to conclude if the peptide is entirely helical or entirely beta sheet conformation. So as per the 4 statements, you need to know the exact function played by the Ramachandran plot and exact function played by circular dichroism. We know that the circular dichroism provides us information regarding the conformation of a protein. or determination of the conformational changes of a protein due to the interaction of any asymmetrical molecules like protein-protein interaction, protein-DNA interaction, protein-ligand interaction, and DNA-ligand interaction. It also helps in determination of thermodynamics of folding and unfolding of proteins as well as nucleic acids. So particularly the CD can tell us whether an amino acid sequence is going to become an alpha helix and beta sheet, and that same data can also be obtained from the Ramachandran plot based on the phi and psi angle. Now if the results are same, then we are going to get the same result from both the Ramachandran plot as well as from the CD spectra. We are not going to see a totally different result from those two. So option 2 is not correct. It says the not possible for phi and psi values to be distributed in the helical as well as beta sheet. No, it's not also true, because we know that the different value of phi and psi may either may either allow the amino acid to form the alpha helix or to form the beta sheet. So both are possible. So the option obviously between 3 and 4. 3 is possible to conclude that the peptide is composed of entirely D amino acids. Now the thing is, yes, this is a possibility, because Ramachandran plot can not only help us to find out whether the phi psi angles, and also the CD spectra, if you think about that the CD spectrum can also provide us important information regarding whether the amino acid the structures are, how the amino acids are arranged. But particularly for a Ramachandran plot, we can easily find out whether it is made up with L amino acids or R amino acid, and that is going to give us information regarding whether a protein is with D amino acids or not. So that's one thing. And the fourth one, not possible to conclude if the peptide is entirely helical or entirely beta sheet. It's also wrong, because that's exactly the job of Ramachandran plot. So Ramachandran plot can do that quite well. So the only thing that is here possible is 3. The question is needed to know the drawback of the Ramachandran plot, and we know normally we find out the possible secondary structure of the protein and R and L characteristics of an amino acid that are present in the protein with the help of a Ramachandran plot. So the drawback is something whether they conclude the peptide is of entirely made of with D amino acid. Yes, a Ramachandran plot can easily find it. Now we are going to see another question. The following are the statements about the pyruvate kinase PK, and there are statements. 1. ATP is allosteric inhibitor of PK. B. Fructose 1,6-bisphosphate is an activator of PK. C. ADP is an allosteric inhibitor of PK. D. Alanine is the allosteric modulator of PK. Now this is nothing but the question from the metabolic regulation, means how a metabolic process in the cell is regulated. And if you see this very closely, you can get to answer this quite fine. See that the ATP is an allosteric inhibitor of PK. So now, as per our discussion earlier in the class, I told you that always this kind of regulation of metabolism. To answer this question, you need to know two things. What are the substrates? What are the products? And you also need to know whether the pathway leads to the production of ATP or consume ATP. If it produces ATP, then ATP will inhibit the process. If it consumes ATP, then ATP will influence the process. Okay. On the other hand, in any cases of this biochemical regulation, the presence of substrate will inhibit and negatively influence. The presence of substrate will always, positively influence, which is also known as modulation. Modulation word is used for a positive influence. Okay. So presence of substrate positively influence. Presence of product inhibits or negatively influence. Okay. So now you need to know this enzyme pyruvate kinase. What is pyruvate kinase and what pyruvate kinase do? Pyruvate kinase is an enzyme that converts phosphoenol pyruvate to pyruvate. So in this step, we know once we are producing pyruvate, we will be producing acetyl CoA. So ultimately we are aiming to produce ATP at the end. So presence of ATP will inhibit this step. Okay. So it says ATP is an allosteric inhibitor of PK. That is true. So if that option A is true, then we know that option C is not correct because A is not present. Statement A is not present. So now among 1, 2 and 4. Let's see. Fructose 1,6 bisphosphate is an activator of pK. Whether it's true or not. Now the thing is, fructose 1,6 bisphosphate is further upstream. Fructose 1,6 bisphosphate is present further upstream. And then further downstream phosphoenol pyruvate is produced. So if more fructose 1,6 bisphosphate is present, so there should be like it's work like a substrate, okay? The process should be like clearance of the substrate, so it will positively influence the process. ADP is an allosteric inhibitor of pK. Now the C statement, ADP is an allosteric inhibitor of pK. Now as we know that this step of conversion of PEP to pyruvate needs 2 ATP, and I mean ATP is converted to ADP is converted to ATP, and actually it's a process, it's a step that generates energy. Okay, it's the last step of glycolysis that generates ATP. So 2 equal process, so 2 ATP produced from 1 molecule of sugar in this step only. So ADP's presence can produce ATP, and ADP's presence is needed. So presence of ADP is positively influenced. But in this case stated that ADP is an allosteric inhibitor of pK, which is wrong. So any option with C is wrong. So 1 is wrong. We know A and B are correct. So obviously D should be correct because there is no option left rather than 2. So if you look at here, you can see this process very clearly, that this conversion of phosphoenol pyruvate to pyruvate, ATP inhibits, alanine, which is further downstream product, inhibits, upstream product fructose 1,6 bisphosphate positively influence, and ATP negatively influence, okay, because ATP inactivate pyruvate kinase from an active state to the inactive state, so it inhibits that, okay. So ATP is an inhibitor, ADP produced to ATP, so ADP is a positive influencer of this pathway, okay. Now the last question, a practical class was going on the students were demonstrating ATP synthesis in vitro using an active mitochondria. Some students added one of the following to their tube. So 4 different statements are provided afterwards. So we are talking about the ATP synthesis in vitro in mitochondria. That means which topic? Electron transport chain. So if you prepared electron transport chain very well, then you probably can answer this, okay. So some added dinitrophenol, DNP, which is an uncoupler of the electron transport. Some added mild acidification, like some cause mild acidification of the medium. Some causes like glutathione that permeabilize both membranes of mitochondria. An outer membrane permeable proton quencher compound, ELILA. So 4 different processes conducted with different tubes. Which of the above, among which of the above tube ATP synthesis will occur or will be detected? That means for the synthesis of ATP, what we need? We need a healthy mitochondria. Let me show that. This is the healthy mitochondria we are seeing. And in a healthy mitochondria, we must have these 3 different compartments, you know. We have a matrix in the center, we have the inner membrane, and we have the outer membrane. Now, dinitrophenol is an uncoupler of the electron transport. That means it will not allow the electron transport chain to continue. So, it will not cause the synthesis of ATP. Because for the synthesis of ATP, the completion of the electron transport chain is very, very necessary. And because during the process of electron transport, the protein complexes that are present and embedded in the inner membrane of mitochondria pump proton from the matrix into the intermembrane space of mitochondria. And the proton gradient which is created in the intermembrane space used as a proton motive force down the concentration gradient to rotate ATP synthase enzyme to produce ATP, right? So we need to keep both inner and outer membrane of mitochondria active and alive. But glutathione permeabilize both membranes. So obviously C will not give us any result of ATP synthesis. And if we allow a proton quencher, that means a molecule or something that's going to quench means associate and attach with the protons and take out all the protons from the environment, then there is no proton gradient form, no chance of ATP synthesis. This is also not going to work. So the only statement which makes logic in mild acidification of the medium, because if we mildly acidify the medium, then that means the cell is under the medium which contains a lot of protons. So the proton will take entry inside the cell, and finally the proton will take entry in the intermembrane space of, normally the protons will be transported inside the matrix, and then that can be pumped into the intermembrane space. So as more protons are present in the environment, more protons will be inserted into the intermembrane space, and as a result, proton gradient, much higher proton gradient can be formed, and more ATP synthesis can be observed. So that is the only way the protein synthesis will be observed among all the 4 examples. So you see exactly from which region the questions are asked. This is very, very important, that when you read a question, try to find out the anatomy of the question, understand every single detail of the question, so that you get to know exact point that is needed to answer the question. If you don't know that exact point, if you don't prepare that earlier, so leave that question there. It will also save a lot of your time during CSIR UGC NET exam. Yes, the answer is yes. If you ask yourself this question, that can you qualify CSIR NET Life Science exam within 6-month preparation? Yes. And if you don't need to believe me, just see the results that our students are getting who joined Shomu's biology coaching for 6-month period of time.
So I'm really thankful for Shomu's biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir, always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is all because of the online test series.
In-depth knowledge what Shomu's biology gave me. I want to thank Shomu biology. I want to thank each and every member of Shomu's biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIR NET GRF journey.
It really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomu sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training, with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIR NET Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us, or simply contact the number given in this video.